All right. Well, welcome everybody who's joining us for the Pediatric Brain Tumor Consortium update. This is an update to focus on the recent spring meeting and some of the clinical trials that are before the Pediatric Brain Tumor Consortium. For those of you that are not familiar with the Pediatric Brain Tumor Consortium, this is an organization that was created back in 1999. So this year is actually the 25th year anniversary for the PBTC. And what it was is it was an initiative that was really done in conjunction with the NIH at the time to focus on effectively centers that were some of the best centers in the country for pediatric brain tumors, and then to have them unify together behind some trials and some new innovations in the pediatric brain tumor space. This has continued, and these organizations periodically go into a competitive type of system, and those best hospitals continue to represent. Today, there are 15 member hospitals of PBTC, both in the United States and also now in Canada. So Dr. Ira Dunkel is the head of the Pediatric Brain Tumor Consortium, and we're glad that you're here to go ahead and give us an update on some of the trials and some of the innovations from the recent meetings and beyond.
Well, great, Keith. Thank you for the introduction, and thank you for inviting me to meet with you and your colleagues today. Keith already sort of introduced this, so I'll just go through this quickly. But when the PBTC was being formed, there was no competing pediatric brain tumor clinical trial group. PNOC did not exist and CONNECT did not exist. So the National Cancer Institute, the NIH, formed this group with the idea that progress was lagging behind in pediatric brain tumors versus some other types of pediatric cancer. And they hoped that bringing together a number of high volume pediatric brain tumor centers with specialized expertise would facilitate doing innovative clinical trials. And as Keith said, there are now 15 sites in the US and Canada, plus the National Cancer Institute as a special site. And the PBTC is intended to do initial phase 1, perhaps phase 2 studies of these new approaches. And then for things that appear promising, we work very closely with the Children's Oncology Group, with COG, the larger North American clinical trials group, and offer our promising strategies to them to do larger scale, more definitive studies. And PBTC is intended, of course, mostly to have specialized expertise, but we also try to have geographical diversity and to cover the large population centers in the United States. And obviously there are gaps where there are no PBTC sites, but these are the current centers geographically distributed. Before I get into our current trials, I thought I'd just very briefly tell you how these trials come to be. So there's multiple pathways for how the trials that we do are initiated. And the most common is that doctors or scientists at the PBTC hospitals bring ideas to the group, and they are constructively criticized and tried to be made better in development process. But also ideas can be brought to us from pharmaceutical companies, from CTEP, which is the branch of the National Cancer Institute that funds us, or from physicians that work in hospitals that are not part of PBTC. So this has happened recently with physicians from the University of Alabama, Birmingham, and with Roswell Park Cancer Center in Buffalo, New York. But regardless of how the ideas are brought to us, they are very thoroughly reviewed by the PBTC Scientific Committee. And if they are approved by the PBTC Scientific Committee, they go to the National Cancer Institute, to CTEP, for a secondary review. And if CTEP approves it, it then comes back to PBTC for us to develop the full-fledged clinical trial protocol. And then that full protocol has to be reviewed and approved again by CTEP, by the National Cancer Institute's Pediatric Central IRB, and in most of the cases, when these are non-authorized medications, by the FDA as well. So it is a pretty rigorous process, and many proposals that are brought forward don't get approved at various stages of the process. So in the 25 years that PBTC has existed, we've completed 42 therapeutic studies. We currently have 9 other studies that are open. In total, we've enrolled just over 2,000 patients onto the studies. And besides the therapeutic studies, we've had 12 non-therapeutic studies that are studying tumor tissue and various biological assays or special radiology studies, things like that. Okay, so I'm going to divide the summary of our clinical trials into different disease types. And I don't know many of you, and I'm imagining that there's varying degree of expertise and that some of you may be extremely expert in a given tumor type, DIPG or medulloblastoma, but perhaps others are less familiar to you. So I'm going to do my best to make this clear, but please, if need be, please feel free during my talk or at the end of the talk to ask for clarifying questions or to make comments. And maybe Keith, you can help with that.
I can. I'll monitor chat if people have any questions that they want to put there, and then I can follow up with questions at periodic points.
Thank you very much. Starting with low-grade gliomas, I think this is probably less of an interest to the Cure Starts Now group, but I thought I would start with this for 2 reasons. One being that low-grade gliomas in children are the most common type of brain tumor. And even though they're not very commonly life-threatening, they are definitely quality of life-threatening because they can affect vision or strength or coordination. And many of these patients need to get multiple courses of surgery, chemotherapy, radiation therapy before they're ultimately cured. The other reason I wanted to mention this is that this is kind of an example of the success that we really aspire to in our clinical trials. So PBTC29 was first a phase 1 and then a phase 2 study of a medication called selumetinib, which is a MEK inhibitor, a type of targeted therapy that's taken by mouth. What prompted the study was the discovery that most children's low-grade gliomas were caused by mutations in the tumor cells in either the BRAF gene or because they had NF1, neurofibromatosis. And we were kind of very lucky because these abnormalities were also responsible for many or most cases of melanoma, the aggressive life-threatening skin cancer in adults. And at this time, there really were no good medical therapies for melanoma. So pharmaceutical companies invested time and money in developing various medications to improve the treatment of melanoma, which turned out to be quite successful. But because the pediatric low-grade gliomas had similar genetic abnormalities, we were able to access the medications as well and use them for children with low-grade gliomas. And in this study, when we did the phase 1 study, where we're looking for the appropriate dose and looking for safety, we saw that many or most of the children's tumors were shrinking, which is very uncommon in phase 1 studies. And that prompted this phase 2 study, which has been completed and just the final papers are being written up. But the data certainly suggests that selumetinib appears to be at least as good and possibly better, easier, safer than chemotherapy for these children. So after this phase 2 study, these data were shared with COG, and they currently have 3 open phase 3 studies comparing selumetinib against standard chemotherapy for these children. So this is really what we aspire to. We do the study, we're clearly helping the children. We can export the strategy to the Children's Oncology Group for a really definitive study. The current study that we're doing for low-grade gliomas and up to a limited degree for high-grade gliomas as well kind of piggybacks on this strategy. So although these targeted therapies work well for a large proportion of the patients, they don't work for everyone. And when they work, they don't always work forever. So Lindsay Hoffman and Jean Mulcahy in Denver had laboratory research and very anecdotal clinical data saying that you could add another medication, hydroxychloroquine, which is not an anti-cancer medication. It was designed as a malaria medication and a arthritis medication, but that it could make the tumor cells more sensitive to the targeted therapy. So we did a phase 1 study seeing if it was safe to add hydroxychloroquine, and it was. And we are now doing 4 phase 2 studies in various types of low-grade and high-grade gliomas to get an early sense of the efficacy of this combination. And one of the phase 2 studies has enrolled all the patients, and the other 3 are near completion. So we don't have results to share with you as to how well this is working, but of course we hope that this will help for the children, young adults who have gliomas that are not responding perfectly to the targeted therapy. I think that most of you in this audience are probably much more interested in DIPG, DMG, high-grade gliomas. So I'll go through some of our efforts there next. So the first study to tell you about comes from Dr. Michelle Monge from Stanford University, who you probably know is an extremely high-powered scientist as well as a clinical pediatric neuro-oncologist who's made a lot of important laboratory discoveries regarding DIPG and high-grade gliomas. And in this paper from a very important scientific journal from 2017, she discovered that rather than the standard approaches that have been used to try to treat DIPG, a completely novel approach of trying to interfere with the normal nerve cells communicating with the DIPG tumor cells appeared to be quite important. And we were lucky enough that a drug to do this had been invented and developed by the pharmaceutical company Incyte for a different reason. They studied this for women in breast cancer. And the drug was not successful for women for breast cancer. So they put it aside. But when Dr. Munjay made her discovery and we approached Incyte, they very generously agreed to give us access to the medication and to make more of the medication for our clinical trial. And I guess, for those of you who don't, I'm sure none of you work in this space regularly, but, sometimes I think people have a sense that there's a antagonistic relationship, or at least a not very cooperative relationship between pharmaceutical companies and hospitals treating children. And I guess I just want to tell you that that's really not my perception at all. So in this case, Incyte really had no financial reason to wish to help us. And in fact, producing the additional medication for the trial was going to be extremely expensive for them. But, they saw it as part of their mission to try to help children with brain tumors. So they really were very generous and provided the drug to us for this study. So the study completed accrual. All the planned children were enrolled to the study. The data is currently being analyzed, and we hope that a paper will be published later this year. And the every other year big pediatric brain tumor meeting, ISPNO, will take place in Philadelphia in July this year. And Dr. Lish Lenzen from Chicago, part of the study team, will be giving a presentation about the data. So this will be our first peek at the data, and they're presented at the ISPNO meeting. A second study that included patients with IPG and high-grade gliomas has been run by Dr. Ralph Saloom at Nationwide Children's Hospital in Columbus, Ohio. The idea here is that a small subset of these tumors are being stimulated to grow by a gene called c-Met. And again, c-Met medications were being developed for other reasons. So Dr. Saloom proposed using one called savolitinib to treat children with brain tumors. So this study is also near completion. We found the appropriate dose of the medication. We've analyzed how much of the medication is in the children's blood after they take it and whether it seems to be enough. And we're currently enrolling patients who have only tumors that have c-Met abnormalities, would be expected to be the most likely ones to respond to the medication. And we've enrolled 6 of the planned 8 patients, so hopefully we'll treat the last 2 soon and be able to report to you and the community whether this strategy is promising. I developed and ran a study using a medication called APX-005M, which is a form of immunotherapy. So this is a medication that's given intravenously every 3 weeks and is intended to stimulate the patient's own immune system and to allow their immune system to more efficiently kill their tumor cells. And this has really been developed primarily for adults with pancreatic cancer and other dangerous solid tumors. But again, the company was very generous and provided us with the drug and some funding to test this in children. So we started looking at children with recurrent malignant brain tumors other than DIPG. And then once we determined safety and a safe dose, We then treated a series of children with DIPGs who had completed radiation and whose tumors had not yet started growing again. This study has also completed accruing all the needed subjects. We are finishing the biological analyses from the blood and the tumor tissue that we obtained from the patients. And this will also be presented at the ISPNO meeting in July. A study that is still enrolling patients, but is getting close to completion, is being run by Dr. Jean Wang at Children's National Medical Center in Washington, D.C., using another immune-stimulating medication called pembrolizumab. Pembrolizumab is clearly effective for melanoma and lung cancer and stomach cancer and many other adult tumors, and is clearly effective for some pediatric cancers like Hodgkin's disease. But when we started this study, it was not clear that it was going to be effective or not for children with brain tumors. Subsequently, doctors in Toronto found that a small subset of children with high-grade gliomas that have lots of mutation in the tumors, these so-called hypermutant tumors, responded very well to this type of medication. And in this study, trying to see whether it will work for other high-grade gliomas as well, the study is divided into two stages. So you enroll a certain number of children, and you have to see benefit in a pre-specified number of them to decide that it's promising enough to continue to enroll patients to the second stage. And this was successful in the first stage. We did see sufficient numbers of patients having tumor shrinkage that this was expanded to the second stage. And we are trying to enroll, I think, the final 5 or 6 patients to the study to determine how effective is pembrolizumab for children with recurrent high-grade gliomas or for hypermutant tumors. Or a completely different approach, Dr. Stuart Goldman, who was at Lurie Children's Hospital when he started this study and has subsequently moved to Phoenix Children's Hospital, and Dr. Chris Tinkle, who's a radiation oncologist at St. Jude, have developed this study of the Optune device. And this device is something that's kind of like EKG leads placed on the chest, but these are placed on the scalp, and it's connected to a battery pack. And unlike EKGs that are measuring the electrical activity of the heart, this is actually treating the tumors with electricity coming from the battery pack to these leads. So it really sounds like science fiction, and probably, many of you are skeptical that this could possibly work. And honestly, I think the medical community was very skeptical as well. But this has gone through phase 3 definitive studies in adults with glioblastoma multiforme in conjunction with standard treatments. And patients who got the Optune device in addition to standard treatment had a better survival than those who did not. Even though it's now an FDA-approved device, it still remains a little bit controversial in adult brain tumor patients, or adult brain tumor doctors, and it hadn't been studied at all in children until Dr. Goldman developed this study. So the study started by treating children with recurrent high-grade gliomas, really to see mostly, is this safe? Can the children wear the device? Because it is a burden. It has to be used for at least 18 hours a day, and it has to be used every day or almost every day. But what we found is that it was feasible, that this was tolerable in children 5 years in age and older. And that prompted Dr. Goldman and Dr. Tinkle to recently modify the study. So it's now available, just recently, treating children with DIPG in conjunction with upfront radiation therapy, and then for the Optune device to continue post-radiation therapy. And the aims of the study are to determine that this is safe in conjunction with radiation and tolerable, and then also to look at the survival of the children and to compare it to the historical controls that we have from PBTC and COG, and hopefully to replicate what was seen in adults with glioblastoma and see that this is extending survival. And then one more study, or another study for children with high-grade gliomas and in the future DIPG, as well as medulloblastoma and ependymoma, is being run by Dr. Claire Twist from Roswell Park Cancer Center in Buffalo, New York. And this is a study using a vaccine trying to stimulate the patient's own immune system to see this molecule survivin as being something that it should attack. Survivin is present on most brain tumor cells, but thankfully is not present on most normal cells. And in adults with glioblastoma, the data from a phase 2 study where this vaccine was added to standard therapy looks promising. And what this curve shows is this is time from enrolling on the study in months. And this is the proportion of patients who are doing well. So 100% were alive and tumor was not growing at the beginning. And then this top curve is the survival of the patients. And the lower curve is survival without the tumor growing. And, you know, of course, this is a little bit glasses half full, half empty, but adult glioblastoma is a very aggressive, very rapidly fatal disease. So these data, while far from perfect, look very promising and are the impetus for studying this subsequently in a randomized phase 3 study in adults, and were the impetus for Dr. Twist and her colleagues to propose this trial to the PBTC as the first trial of this vaccine in children. So children get the vaccine as an injection, like any other vaccine, every 2 weeks initially for 4 doses. And then I think it's given every 2 months after that for as long as it's working. We started with older children to make sure it didn't appear to be dangerous. When that appeared to be safe, we opened it to children with recurrent tumors who were younger children. When we have one more child assessed and we hopefully confirm that is safe, we will then open a third stratum, a third arm of this trial for patients with DIPG after radiation therapy. Then the final trial I want to tell you about for high-grade gliomas is one that was brought to us by Dr. Greg Friedman, who initiated this when he was in Birmingham, Alabama, although he's recently moved to MD Anderson Cancer Center in Houston, Texas. And this G207 is a form of the herpes virus that causes cold sores in our mouth commonly, but it's been engineered in the laboratory so it can't infect normal cells and make people sick, but it can infect tumor cells and kill them and cause inflammation and bring an immune system attack on the tumor cells. This medicine is given with injections directly into the tumor. So the patients go to the operating room, they have 1 to 4 catheters placed in the tumor, they go to the intensive care unit, and then the next day they get a several-hour infusion of this oncolytic virus. And then the next day they get 1 dose of radiation that's intended to help the virus do its job in the tumor cells. And from the initial study that Dr. Friedman did in Alabama, these are MRI scans looking at the brain from the side. So here's the eye, the back of the head, top of the head, bottom of the brain. And these are 2 different sequences on the MRI scan. So it's the same patient, just different time points, looking at the tumors in 2 different ways. And the tumors, the white part here, or the white part here, this would be tumor and perhaps some swelling around it. And what you can see is that after the injection of the virus, over time, the tumor shrinks and develops what Dr. Friedman calls a Swiss cheese pattern. So even within this residual abnormality, it looks like the tumor is liquefying and dying. But this process can take an extended period of time. So because of this patient's promising result and some others that Dr. Friedman treated, we are now conducting this phase 2 study where all the children will get the same dose of the virus. And the metric of the study is to look at survival of these children with recurrent high-grade gliomas at first recurrence compared to matched patients from prior COG trials that are very similar to the patients that are being enrolled on this study. This patient just very recently opened to accrual. Last week, the first hospital received authorization to start treating patients. And I think the first patient is being assessed this week as a possible candidate. Okay. For medulloblastoma, much more briefly, Dr. Saloom also has a study for children or adults in this case with a subtype of recurrent medulloblastoma. And this is based on preclinical data from a laboratory at Stanford University showing that this appeared to be promising in the laboratory. This is a targeted therapy that interferes with this pathway that's turned on when this gene is mutated in these tumors. And there are separate strata for children who are still having bone growth because there's some concern this medication might affect bone growth. Older children and adults who are skeletally mature, excuse me, I had that backwards. And a separate one for patients who can get medication and then get a clinically indicated surgery so we can measure exactly how much of the medication is getting into the tumor cells. The other medulloblastoma study I wanted to very briefly tell you about is this study being led by Dr. Karianis at my institution, Memorial Sloan Kettering. And this is a study that is adding this investigational medication to standard second-line treatment with these chemotherapy medications for recurrent medulloblastoma. And the question is, we have data from COG about how well these medications work. We have preliminary data from Sloan Kettering suggesting promise of this medication by itself. We want to know if adding this to the standard therapy will improve the prognosis for children with recurrent medulloblastoma. This also kind of tells a story that I just wanted to briefly share with you about the pitfalls of clinical research. So this study opened, and there was a lot of enthusiasm among patients and parents and physicians, and children were being referred quite regularly for this study, and we were accruing quite well. But the company from Denmark that's developing this medication was hoping to get approval for this for a different disease. And the FDA in late 2022 declined to approve the medication without additional studies being done. So the company kind of temporarily ceased producing this medication until they sorted out among themselves and with the FDA whether they would continue to develop this by doing the other clinical trial or trials that the FDA wanted. And unfortunately for us and for the medulloblastoma community, because the drug was no longer being made for the other disease, we lost access to it also for medulloblastoma. So for a little bit more than a year, we've been unable to treat any patients on this study. And we expect to find out in the next month or two whether the drug will become available again and we can resume the study, or whether the drug will not be developed any further and we will have to close this study. Okay. For ependymoma, just very briefly, we have this one study I wanted to tell you about being run by Dr. Hegde at Texas Children's in Houston. CAR cells are patients' own T cells, own immune cells that are taken from the patient, altered in the laboratory to attack tumor cells, and grown to large quantities, and then given back to the patient. So in this case, the CAR cells are altered or engineered to attack something called HER2, which is commonly on the surface of ependymoma tumor cells. Dr. Hegde had collaborated with a very prominent neurosurgeon researcher, Michael Taylor, when he was in Toronto. He subsequently has moved to Texas Children's and showed in the laboratory these cells appeared to be quite effective at treating ependymoma. And that prompted us to open this study of these cells in children with recurrent ependymoma. CAR cells, I should have said perhaps previously, have been a major breakthrough for children and adults with blood cancers. But whether they work or how often they work for children or adults with brain tumors or other solid tumors is really uncertain. And there are active research programs at Texas Children's, at Seattle, at Stanford University, other places, trying to develop CAR cells for brain tumors and other solid tumors. But what's unusual here is that usually those are studies where patients have to go to Stanford, have to go to Seattle, and there's a burden on the child and the family traveling and staying in this other city, et cetera. For this study, the children stay at the PBTC site. They have their blood drawn locally at their hospital. The blood is sent to Texas Children's. The cells are created at Texas Children's and then shipped back to the PBTC sites. And then the children get some mild to moderate chemotherapy that aids the CAR cells surviving in the patient's body. And then these CAR cells are given to the patient. And this can be repeated up to 3 times if it's working, if it's safe. So this study is trying to see that this first academic multi-center study is feasible and safe. Of course, what we really want to know is whether it appears to be helping the children. And we've treated a number of children. The study is going well, and this study is expected to remain open for future patients for the next few years. Then the last thing I'll tell you about is one non-therapeutic study that we have, but with big therapy implications potentially. And this is a study of cell-free DNA being analyzed in the CSF, the fluid that's made in the brain and can be obtained from lumbar punctures or from other procedures. This is being run by a neuro-oncologist, Dr. Alexandra Miller, who until recently was at Memorial Sloan Kettering and just moved to New York University. And the idea here is that in recent years, it's been shown that tumors can shed DNA into the blood. And brain tumors, it's hard to find in the blood, but it has been found that in a substantial proportion of patients with brain tumors, you can find this tumor DNA in the CSF fluid. So the idea here is that we would like to test tumor tissue and CSF and see how similar are they. Does the CSF sample tell us more or less the same thing we can find from the tumor tissue, in which case this would be a much safer way of biopsying to make a diagnosis and to get this genetic information. This is also something that can be done much more safely repetitively, so we would never subject children to multiple tumor biopsies over time. But if this is valuable in providing information following the children, it's a relatively minor procedure to do the lumbar puncture. And this may be a way of complementing MRI scans to monitor the response to treatment or to monitor for recurrence beyond the MRI scans, which is really our only tool right now. And I think the other thing that's interesting about this is that this is not purely a research test in the sense that if patients and doctors enroll their patient onto the study and send this CSF sample to Memorial Sloan Kettering, unlike most research studies done in a laboratory where the patient doesn't get the results back, the results of this analysis are provided as a clinical test to the referring doctor to share with the patient. And this information can be used based on the best judgment of the doctor for clinical decision making. So this study opened, it's actually on hold for the moment because we have to make a minor administrative change, but should be up and running again in the next couple of months. So I'm sorry if I perhaps went a little longer than I was expecting, but I just want to thank everybody at PBTC, the other doctors, nurses, clinical research teams, Dr. Arzu Onar-Thomas, who's the co-leader of PBTC based at St. Jude, Jason Fangasaro, who's my vice chair, and of course, especially the patients and their families who've enrolled their themselves or their children on PBTC studies. And thank you to you all and Keith for all the work that you're doing supporting pediatric brain tumor research.